Welcome to the lecture series on photosynthesis, which will focus on a detailed aspect of the Hill reaction or the light reaction. This is divided into two parts for ease of comprehension. Before beginning, I wish to acknowledge the research publications that have provided me with the details I share here. Photosynthesis performed by modern plants has its evolutionary origins in cyanobacterial systems, which have further shown to have evolved from those of the anoxygenic phototrophs like bacteria and archaea. Chloroplasts in plants, where photosynthesis is performed, have evolved through the endosymbiosis of cyanobacteria with eukaryotic cells over a billion years ago. The high degree of conservation observed especially at the central region of photosystems 1 and 2 between plants and cyanobacteria suggests the role of strong constraints that act upon the photosynthesis process, preventing further changes to the pigment geometry. Due to the large difference in the redox potential between the electron donor and final electron acceptor during the light phase of photosynthesis, the ancestral cyanobacteria had to evolve the capability to use two photosystems working in series in order to be able to accumulate the energy of two photons. An ancestral symbiotic event between a cyanobacterium and a eukaryotic cell transformed the first organism into a protochloroplast and opened the way to evolution of green eukaryotic photosynthetic organisms, while other subsequent symbiotic events also allowed the evolution of other types of eukaryotic photosynthetic organisms. The first land plants had to challenge new environmental constraints and stresses: an atmosphere rich in oxygen, an environment rapidly fluctuating in terms of light quantity and quality, temperature, nutrients, and water. In green organisms, most of the light is absorbed by the photosynthetic pigments chlorophyll A and B, which have remarkable physicochemical properties, allowing efficient light harvesting and ultra-fast excitation energy transfer amongst antenna chlorophylls. Whenever a chlorophyll molecule gets hit by a photon, one electron gets excited to a higher orbit and releases very high energy when it returns back to the ground level. This energy can be detrimental to the pigment and other biomolecules associated with the pigments by way of oxidizing them, thereby affecting the photochemical conversion, thus stalling the metabolism. This wastage of energy is effectively controlled by the electron acceptors that are assembled in close proximity to the reaction center that function as electron transporters, thereby transforming the energy to synthesize ATP and NADPH, the assimilatory powers for all anabolic reactions in the living system. This photochemical conversion has been highly conserved through the evolutionary ages, with some changes incorporated in the integration of the pigments in the form of tightly or loosely adhering proteins and lipid molecules, thus forming supercomplexes and megasupercomplexes. The similarity in composition and arrangement of cofactors in complexes that are separated by a billion years of evolution is striking, and the differences in protein-protein interaction and new features added during the long-term evolution of eukaryotic complexes emphasize the unique biological adaptations to ecological niches and environmental variation of these complex enzyme systems. These are the overall photochemical reactions taking place in the plant chloroplasts, leading to the key results like photon-induced excitation of electrons from the chlorophyll molecule, oxidation of water to produce oxygen, effective removal of the electrons and protons liberated during the oxidation and excitation processes via a set of electron transporting cofactors in a series of oxidoreduction reactions. And finally, the electrons are being absorbed by NADP to produce the high energy assimilatory powers, the biological hydrogen molecule, as well as to channelize the protons to produce ATP via the enzyme complex ATP synthase. There is a discrete spatial organization for the attachment of the protein enzyme cofactor complexes to the thylakoid region of the chloroplast. The assembly for photooxidation of water and synthesis of ATP NADPH are part of the Hill reaction or light reaction that is found organized in the grana portion as well as in the stroma lamellae of the thylakoid, both embedded in the membrane as well as on the lumen side. The assembly for the assimilation of NADPH molecules and ATP molecules, known as the dark reaction, leading to the reduction of carbon dioxide via the RUDP molecule to various sugars like sucrose and glucose, is organized in the stromal matrix of the chloroplast. The light reaction proceeds at a very fast rate from the time a photon hits the reaction center to the excitation of electrons and transfer of electrons. The whole process of one transfer gets over within microseconds. The charge separation step within the reaction center is the rate limiting step. A photosynthetic unit is a group of about around 230 chlorophyll molecules and other pigments which take part in photochemical reaction, that is the light reaction. Such a unit is called a quantazome. They are found in the unit membrane of granum disk of chloroplasts. Chloroplasts always contain both chlorophyll A and chlorophyll B. Most plants contain about twice as much chlorophyll A as chlorophyll B. Chlorophyll is always associated with specific binding proteins forming light harvesting complexes in which chlorophyll molecules are fixed in relation to each other, to other protein complexes, and to the membrane. Chlorophylls A and B and bacteriochlorophyll are the primary gatherers of light energy. Phycoerythrobilin and phycocyanobillin are the antenna pigments in cyanobacteria and red algae. Beta-carotene and lutein are accessory pigments in plants. The light-absorbing pigments of thylakoid or bacterial membranes are arranged in functional arrays called photosystems. There are two photosystems in plants and algae that are the seat of the light reaction or the oxygenic photochemical conversions. The two photosystems have a common organization and are functionally organized in two main moieties: a core complex containing the reaction center where the photochemical reactions occur, and a peripheral antenna system that increases the light harvesting capability. These accessory pigment systems are directly involved in the photochemical conversions as well as in regulation of the photosynthetic process. While the synthesis of the reaction center complex requires substantial energy, its effectively absorbing cross-section is very small. Phototrophic organisms therefore employ accessory protein components, the antenna or light harvesting complexes that use sets of cofactors to absorb photon energy and funnel it to a limited number of interspersed reaction centers. This has given rise to the massive photosystem complexes 1 and 2. Photosynthetic reaction centers are most commonly heterodimer pigment protein complexes, and this is the case for all known oxygenic photosystems. First of all, a clear difference exists between the pigment composition of the core complex and the external LHC antennas. The core complex of both the photosystems bind essentially only chlorophyll A and carotene molecules, while the external LHC antenna complexes also bind chlorophyll B and xanthophylls, which are oxygenated carotenoids. There are linker chlorophyll pigments referred to as accessory chlorophylls, and they are found in close association with the RCs, molecularly referred to as P680 FeO in Ps II and P700A0 in the case of Ps1. The co-complexes have been well conserved during the evolution, as most of the subunits are similar in prokaryotic and eukaryotic photosystems, and only a few are specific to each group. On the contrary, the peripheral antenna system displays great variability, being composed of peripheral associated membrane proteins in cyanobacteria, the phycobilisomes, and integral LHC membrane proteins in eukaryotic cells. The photosystems are connected by an intermediate membrane supercomplex called cytochrome B6F and two electron carriers, one liposoluble quinone molecule, plastoquinone, that transports electrons between the photosystems, cytochrome B6F, and the luminal copper-containing soluble protein plastocyanin that links cytochrome B6F to PS1. All the pigment molecules in a photosystem can absorb photons, but only a few chlorophyll molecules associated with the photochemical reaction center are specialized to convert light to chemical energy. The other pigment molecules in a photosystem are called the light harvesting or antenna molecules, where they absorb light energy and transmit it rapidly and efficiently to the reaction center through the resonance transfer mechanism. The primary photochemical act occurs at the reaction center only. The two systems have distinct and complementary functions. Both the photosystems act in tandem, that is, they function simultaneously in series. The reaction center of both the photosystems are special chlorophyll pairs named P680 in Ps2 and P700 in Ps1. These are the first cofactors discovered to be functionally involved in primary electron transfer reactions. It has been generally considered that both P680 and P700 act as the primary electron donors, directly reducing the respective electron acceptors PO-D1 protein complex and P700 A0 protein complex, and hence forming the radical pairs as a result of a phenomenon called charge separation. Ps2 reaction center contains equal amounts of chlorophylls A and B. Ps1 has a reaction center with a higher ratio of chlorophyll A to chlorophyll B. Ps1 and Ps2 are spatially distributed along the thylakoid membrane. The photosystems are so arranged in the thylakoid region that release of electron from Ps2 replenishes the loss of electron from the other Ps1, each one acting in a series continuously. Electron removed from water molecule replenishes the loss in Ps2. Ps1 and Ps2 are spatially separated on the chloroplast interior so as to distribute the energy released evenly, thus preventing damage to the photosystems. If Ps1 and Ps2 were physically contiguous, since the energy required to excite Ps1 is less than that needed to excite Ps2, it may need faster replenishment of electrons, and it being close to the Ps2 will leave Ps2 chronically underexcited and thus affecting the rate of photosynthesis, which is in contrast to the emerson's enhancement effect. This kind of wastage is prevented by separation of Ps1 and Ps2 in the thylakoid membrane. Ps2 is located in the tightly appressed membrane stacks, that is the granule lamellae, and Ps1 is located in the unappressed stroma lamellae. Now we are going to see the general aspects of photosystems. Photosystems contain the so-called reaction centers, which are the sites where the photochemical reactions occur, typically divided into families or groups depending on the chemical nature of the terminal electron acceptors. Photosystem 2 has a quinone type reaction center, also known as the Q type or type 2, while photosystem 1 has an iron sulfur type reaction center. Both families of the reaction centers are present embedded in the thylakoid membranes of the chloroplasts. Photosystems interact with other membrane complexes, such as the LHC proteins or soluble proteins that mediate electron transport. A calculation of the total number of chlorophylls associated with Ps1 and Ps2 suggested that more chlorophylls, approximately 10%, are associated with Ps1 than with Ps2. In agreement with the results showing that Ps1 absorbs approximately 20% more photons than Ps2, since the pigments in Ps1 are more tightly packed than in Ps2. A single plant chloroplast has more than hundreds of both the photosystems. Photosynthetic reaction centers are embedded in the so-called core complexes that are made of a pair of chlorophyll molecules tightly associated with protein molecules, where a phenomenon called charge separation takes place that triggers the downhill transfer of the excited electrons, leading to its energy dissipation. The reaction centers are further surrounded by many protein subunits, forming the light harvesting complex that is responsible for efficient transfer of the photons to the reaction center. Moreover, there may be aggregation of the LHCs depending on the quantity of light leading to the formation of supercomplexes to facilitate maximum trapping of the photons. Sometimes these supercomplexes may further integrate, forming megacomplexes too, to regulate the process of absorption of photons. A group of pigment molecules, collectively called the antenna pigments, also form a part of the LHCs. They are made of pigments other than chlorophyll A or B, and their main function is to absorb photons of all other wavelengths of their absorption spectrum, thereby assuring the efficient use of the most of the solar radiation, passing them to the LHCs, further to the reaction centers where the photochemical conversion occurs. There are two groups of antenna pigments, the peripheral antenna system and the central antenna system. Their function is best comprehended in varied light intensities, since they also have a protective role to play under such very high light intensities. There are a number of cofactors arranged in close association with the reaction centers for efficient transfer of electrons and protons that form the basis of the entire photochemical conversions. The reaction center embedded protein complexes are arranged parallel to the membrane, whereas the cofactors are arranged perpendicular to the membrane. This configuration allows efficient and fast transfer of electrons across the membrane and the formation of a photochemically generated electrochemical potential. The core of PS2 is a multi-subunit complex composed of about 25 to 30 subunits. The plant RC is a dimer surrounded by tightly bound LHC2 complexes. This supercomplex can interact with different numbers of additional LHC2 units depending on light intensity and quality. The two most important functions of PS2 is the lysis of motomolecules triggered by photons and release of molecular oxygen. PS2 utilizes a bound inorganic ion cluster of 4 manganese ions, 1 calcium ion and 1 chloride ion to oxidize water and produce the electrons for reduction of plastoquinone, which functions as the mobile electron carrier, coupled with the release of oxygen. PS1 core has lesser number of subunits when compared to PS2, explaining the need for a lesser energy to excite the reaction center. It is actually observed through valid scientific explanations that the reaction center of both the photosystems are thus composed of very closely associated electron donor and electron acceptors, forming a pair as a result of a charge separation act, which is the existence of a transitional negative and a positive charged ionic state. Formation of this state is crucial for the downhill transport of electrons through the cofactors. It has been generally considered that both P60 and P70 act as the primary electron donor, reducing directly the respective electron acceptors. The photochemical population of P680 FeO and P700 A0 could be thus considered equivalent to the trapping of the excitation energy into a chemical form. The photosystems enter into two different transitional states aided by the LHC2 that is associated with the photosystem 2, depending on the quality and quantity of sunlight availability. At state 1 transition, both the photosystems operate normally so as to absorb maximum photons. At this state, LHC2 is favorably associated with PS2 as a result of dephosphorylation of a threonine residue at the LST2. When there is intense light, that is blue region, which is available, there is a possibility of a state 2 transitional level where the PS2 gets activated faster than PS1 since it is the preferable wavelength for PS2, leading to a faster generation of reduced plastoquinone than its oxidative state. That is to say that PS1 is unable to accept electrons from plastoquinone, reduced plastoquinone at that faster rate, since PS1 does not get enough photons to get excited as compared to PS2. At this time, due to the activation of a kinase enzyme, the threonine residue in LHC2 gets phosphorylated. This results in the dissociation of some LHC2 units from the PS2, which move towards the stroma lamellae and aids to harness extra photons so as to pass it to PS1 and enhance its electron excitation rate. This helps the PS1 to accept electrons from the reduced plastoquinone and normalize the flow of electrons, thus preventing stalling of the light reaction. In less intense light distribution, that is in the shade, PS1 oxidizes the reduced plastoquinone faster than the creation of the reductant. And this triggers the dephosphorylation of the threonine residue in the LHC2 subunits, which leads to its reassociation with the PS2 to regulate the synthesis of reduced plastoquinone, thereby normalizing the transfer of electrons and reaching back to the transitional state 1. Reaction center in the photosystems where the actual conversion occurs is always essentially a dimer or a pair of pigment molecules, normally chlorophyll A or an epimer of chlorophyll A. It is here that the difference is observed between the two photosystems. When the pigment molecule in the reaction center is either hit by a photon directly or through the accessory pigments in the LHC, one energy-rich electron is pushed out of its orbit. The molecule becomes very unstable and it strives to return to its ground state, that is the original state. This can happen in 3 ways. First, if the electron loses the energy, sometimes as heat, sometimes as light. Secondly, the energy of the electron can be passed to a nearby atom. Or thirdly, the electron as well as the energy can be transferred to a nearby atom. Plant pigments usually utilize the last 2 of these reactions to convert the sun's energy into their own. Because the reaction center is arranged in such a way so that a suitable electron acceptor is nearby, the excited electron can move from the initial donor molecule to the acceptor molecule. This process results in the formation of a positive charge on the special pair due to the loss of an electron and a negative charge on the acceptor, and is hence referred to as photo-induced charge separation. This occurs in less than 10 picoseconds. Incidentally, 1 picosecond is equal to 10 to the power of minus 11 seconds. At this stage, the pair could undergo a phenomenon called charge recombination, where the electron could be reverted back to the donor, thus neutralizing its positive charge. This is a waste of energy since it only transforms the absorbed energy to heat. This backflow of electrons in PS2 can produce reactive oxygen species, leading to photoinhibition or photon-induced damage. 3 factors in the structure of the reaction center work together to suppress charge recombination nearly completely. First, another electron acceptor is less than 10 angstrom away from the first acceptor, and so the electron is rapidly transferred farther away from the special pair. Second, an electron donor is less than 10 angstrom away from the special pair, and so the positive charge in the special pair is neutralized by the transfer of another electron from the donor. And third, the electron transfer back from the electron acceptor to the positively charged special pair is especially slow. Thus, electron transfer proceeds efficiently from the first electron acceptor to the next, creating an electron transport chain that ends if it has reached NADP. This remarkable efficiency and directionality of electron transfer in the reaction center is essential for photosynthesis to work, and its basis is the evolutionarily optimized, precise positioning of the cofactors that is almost impossible to mimic in the artificially conducted light-driven charge separation. It is confirmed that productive charge separation will only result if the actual absorption of a photon occurs on the chlorophylls of the special pair. Thus, nature has devised pigment systems and organisms have evolved to efficiently harvest all colors of the sunlight impinging on the earth. This large variety of pigmentation is integrated into a range of functional pigment protein structures referred to as the photosystems. The cofactors participating in the transfer of the excited electrons from the reaction center are closely associated with the LHC and the reaction center for faster transfer of electrons, leading to faster replenishment of the oxidized cofactors that are ready to accept the next set of electrons from the RC. The photochemical reaction center of both the photosystems is composed of 6 chlorophyll A molecules. In case of PS1, all the 6 chlorophyll A form part of the reaction center. Further, there are 2 phylloquinones distributed on either side of the RC. Joining the either side, there are 4 iron sulfur clusters, one FX center, beyond which there are the ferredoxin kind of terminal electron acceptors designated as FA and FB. Whereas in photosystem 2, 4 are chlorophyll A directly involved in the photochemical reaction, and 2 are pheophytin molecules, which are type of chlorophyll A molecules that are associated with protein subunits designated as D1 and D2. Further, the pigments are more tightly packed in the case of PS1. This means that interactions amongst the pigments comprising the reaction center of PS2 are in general terms weaker than that of PS1 RC pigments, and the cofactors in the RC of PS1 form 2 quasi-symmetric branches, that is 2 almost symmetrical branches diverging from chlorophyll A prime pair, traditionally called P700. In each branch is another pair of chlorophyll A molecules and a phylloquinone. Finally, the branches join again at the FX iron sulfur cluster. In the case of PS2, only one of the 2 electron transfer branches is used with high efficiency, A branch, which is called, and therefore only one of the 2 pheophytin molecules bound to the D1 subunit is reduced photochemically. This is known as monodirectional or asymmetric electron transfer. On the other hand, in PS1, both electron transfer branches are used with comparable efficiencies so that both the A0A and A0B are reduced following charge separation. The latter is called bidirectional or symmetrical electron transfer. As a result of the monodirectional electron transfer in PS2 RCs mentioned above, only one of the 2 plastoquinones, that is QA, which is coordinated to the D2 subunit of PS2, is reduced to the semiquinone form directly from Pheo D1. The remaining quinone, that is QB, is reduced to the semiquinone by QA as a result of one charge separation event, that is absorption of one photon, and to the fully reduced quinol form QB2, which is then protonated to become QBH2 after a second charge separation event, that is absorption of 2 photons, which is a process known as 2-electron gate. QB represents the terminal electron acceptor of PS2, which in a reduced and protonated form can diffuse out of the RC binding site and act as a lipophilic electron carrier within the thylakoid membranes. On the other hand, in the case of PS1 reaction center, the phylloquinone molecules coordinated by protein subunits A1A and A1B are reduced directly from the upstream cofactor in the electron transport chains, that is the chlorophylls A0A and A0B. Moreover, the phylloquinones are not the terminal electron acceptors, as their semiquinone form is oxidized by the iron sulfur cluster FX, which is a common cofactor to both the electron transfer chains. The electrons are then transferred sequentially from FX to the other 2 iron sulfur centers associated and catalyzed by the enzyme NADP ferredoxin reductase with the coenzyme ferredoxin, which represents the terminal electron acceptor within the RC. The reduced soluble electron carrier protein ferredoxin further transfers the electron to NADP to produce NADPH. Differently from the case of PS2, where 2 electrons are accumulated by the terminal acceptor QB and 4 oxidizing equivalents, that is electrons, are accumulated at the donor side at the level of the oxygen evolving complexes, so that the system is effectively reset after 4 charge separation events. In PS1, there is no accumulation of reducing or oxidizing equivalents within the reaction center. Hence, the donor side of PS2 is significantly more oxidizing. On the other hand, the acceptor side of PS1 is significantly more reducing than that of PS2. Hence, in both cases, approximately 60% of the energy delivered by a photon is being utilized in the photochemical conversion. The rate limiting step in photosynthesis is the transfer of electrons to the PS1 and transfer of protons across the thylakoid membrane so as to create a gradient difference by way of increased number of protons on one side of the membrane. This is managed well by the intermediary liposoluble dimer molecule, that is the cytochrome B6F. The cytochrome B6F complex, which is referred to as plastoquinone plastocyanin reductase, occupies a central position in the sequence of photosynthetic electron transport carriers, providing the electron transfer connection between the two reaction center complexes PS2 and PS1, to which proton transfer is coupled, thus contributing to the transmembrane electropotential gradient that is the basis for synthesis of ATP. Cytochrome B6F complex has been shown to be a lipoprotein dimer and to contain 8 in cyanobacteria to 9 in higher plants, tightly bound subunits or monomers in the chloroplasts. The extra subunit is the ferredoxin NADP reductase, FNR is as it is referred to in higher plants, that explains the cyclic flow of electrons observed in plants. 3 of the polypeptide subunits involved in the transfer of electrons in the B6F complex are cytochrome F, cytochrome B, both containing heme prosthetic groups, and the risky type 2 iron sulfur protein containing non-heme prosthetic groups. Prosthetic groups are any non-protein component like metal ions that are tightly linked to the enzymes or subunits of enzyme complexes. Chlorophyll A molecule and beta-carotene is also found associated with the complex. The 2-electron oxidation of plastoquinol, PQH2, occur via a split pathway. 1 electron donated to the high potential iron sulfur protein, which is the electron donor to the cytochrome F, the other to the pair of lower potential cytochrome B hemes.